All right, everybody. So here we go to wrap up chapter 18. This is part two of chapter 18, pages 654 to 674. And we're going to be talking about aspects of mitosis here, but not through a genetic lens, of course, through a cell biology lens instead. More specifically, we'll be talking about centrosomes and the mitotic spindles, in other words, the microtubules, and what they do during mitosis, and that includes them making the mitotic spindle. That'll lead us to talking about chromosome positioning and sister chromatid separation there in metaphase and anaphase. From there, we'll go into the mitosis checkpoint, which happens right there at the metaphase-anaphase boundary. And then we'll move on to actin's role. We'll talk about cytokinesis and the contractile ring, how cytokinesis or cell splitting works in plants, and then we'll briefly end with a quick discussion talking about how the cell works to control cell number. So we've, at this point, if we just build on the last video, we've passed through our checkpoints. The cell is fully prepared now to divide and fully cleared to divide as well. All the DNA has been replicated in S phase. Chromosomes have condensed. They're very tightly packaged. They're bundles of DNA. They look like this, so they're not tangled and they won't get all tangled up. That's what prophase is for. And so it's time to separate sister chromatids. It's time to pull those exact copies of the chromosomes, those sister chromatids, to opposite sides of the cell, and then split that one mother cell into two. So what are our challenges here? Well, we have a few. First, the sister chromatids are essentially glued together by the protein called cohesin. Cohesin is a protein that literally forms a ring around the sister chromatids and keeps those sister chromatids tightly bundled together. So in order to get these sister chromatids separated as we transition from metaphase into anaphase, that cohesin must be first destroyed. Cohesin has to be cleaved so that the sister chromatids are no longer bundled in this way, and then the microtubules, the mitotic spindles, can pull those sister chromatids apart. So in metaphase, the sister chromatids are still stuck together, cohesin is still intact, and then as we transition from metaphase to anaphase, we have to destroy that cohesin so the sister chromatids can be pulled apart. Then we make new nuclei by dephosphorylating the nuclear lamins, and we can pinch the cell apart into two cells through cytokinesis. So what we need to cover today to fully understand all of these processes and how they happen in a cell biology perspective and comprehensively cover the cell cycle is first, how do the mitotic spindles assemble and how do they function? This process involves the dynamic instability of microtubules, involves motor proteins walking along those microtubules, and some other concepts we've already covered. How does the third checkpoint work? We mentioned that in the last lecture, where there's this mid-mitosis checkpoint in anaphase. We need to make sure that those microtubules are attached properly. We'll discuss that. And then how physically does the cell actually split into two? We know a little bit about that being this ring of actin that needs to contract and needs to be pulled apart, but we'll talk about that in more detail here too. And finally, how do cells control their overall cell numbers and their overall cell size? This is a wonderful system for cell division, and checkpoints provide the regulation, but can we do something else to control cell division? Is there something we can do to stop cells from dividing uncontrollably, because uncontrollable cell growth is cancer? So let's talk a little bit about centrosomes, how the centrosomes function in order to make the mitotic spindles. The first thing that happens in M phase is the first stage of mitosis. Remember from the last video that M phase is mitosis and cytokinesis. So M phase marks the very official end of interphase and the end of G2. And once we enter M phase, we are beginning mitosis. Therefore, for M phase to even be remotely possible, S phase needs to be long done and all the DNA must be fully replicated. And also to prepare for chromosome segregation, which is what mitosis is for, the centrosomes must also be duplicated because we need centrosomes on either side of the cell so that microtubules can be projected from opposite sides or opposite poles of the cell, and those microtubules can attach the sister chromatids and pull them apart. Remember from our cytoskeleton lecture, the centrosome is the origin point of all microtubules. Microtubules must form at those gamma tubulin rings that are embedded in the centrosome. That's what centrosomes do, is allow microtubules to form and organize. And so if we need microtubules projecting from opposite poles of the cell, we need centrosomes at opposite poles of the cell. And then as those microtubules pull those chromosomes apart, each daughter cell will not only get a full complement of chromosomes, but each daughter cell will also get its own centrosome because the centrosome was there located at opposite poles. So centrosome duplication is important for mitosis, but it's complicated. And so centrosome duplication actually begins at the start of S phase. And it's triggered by, the centrosome duplication process is triggered by, no surprise, the SCDK and the G1S boundary CDK. The same CDKs that get us into replication of the genome also initiate the replication of the centrosome to prepare for the two centrosomes that need to move to opposite sides of the cell right there in the beginning of mitosis. At first, these duplicated centrosomes stay together. But as we transition from S phase into G2 and G2 into M phase, those centrosomes are going to separate. They move to opposite sides of the cell, and they form a structure that we call asters. Asters because in the microscope, back in the early days, in the olden days, these asters looked like stars, stars twinkling in the night. So aster, like astrology, aster is Latin for star. And these were actually the beginnings of the microtubular arrays. The reason why these looked like stars is because they were centrosomes with the beginnings of microtubules projecting out of them. So the mitotic spindle, these microtubules emerging from the centrosome, start to assemble with earnest, start to assemble properly in prophase. One last reminder I'll say. Before we go any further in this video, please make sure that you're comfortable with mitosis. And if you're not, I'm begging you to pause this video and refresh your notes, refresh your thinking, go back to your genetics textbook and become familiar with mitosis. I'm going to teach this material with the assumption that you've got mitosis down. So make sure that's the case before you try to build on that foundation. I'm going to be referring to the stages of mitosis by name, and I'm going to assume that you know what goes on in those stages. All right, so back to this. So the mitotic spindle forms in prophase. That's what happens in prophase. Also, chromosomes are condensing fully and they're becoming tightly wrapped. At the start of mitosis, dynamic instability is in full effect. The anchoring proteins that have kept microtubules intact as the tracks and the roadways of vesicle transport, well, those anchoring proteins have popped and let go. And since all of the alpha and beta tubulin in those microtubules have been holding on to GDP all this time, they are very much unstable, and the microtubule breaks down. That's all happening very early on in prophase. Now, part of what's causing these anchoring proteins to pop and let go of microtubules is that they're being phosphorylated by MCDK. So MCDK is an active kinase. It's active because the M cyclin is made at the beginning of M phase, and it phosphorylates these microtubule anchoring proteins. And so microtubules are no longer stabilized because the proteins that stabilize microtubules have become inactivated through this phosphorylation. And without these anchoring proteins, the mitotic spindles, the microtubules, become less stable. During prophase, now that we're in mitosis proper, the centrosomes start sending out these new microtubules that they're able to make because the previous microtubules have broken down, and they start fishing. We talked about that in the cytoskeleton lecture, but those centrosomes just send microtubules out in all directions, waiting for those microtubules to find some kind of anchoring point. So randomly, in all directions, microtubules are being made. And again, this is what gives these star-like appearances to the centrosomes. This is why they were called asters. A greater number of microtubules are sent out from the centrosome, and the rate of casting out these microtubule lines is also going up. Now, of course, some of those microtubules are going to, by random chance, touch other microtubules that are growing out of the other centrosome. So these microtubules basically anchor to each other and stabilize each other, avoiding dynamic instability. So the first thing that happens is microtubules touch microtubules being projected from the opposite centrosome, and they stabilize and anchor each other. These joined sets of microtubules from opposing centrosomes on either pole of the cell make up what we refer to as the mitotic spindle. The centrosomes are now referred to as spindle poles because they are the poles from which these spindles are emerging, and they anchor the other side of the microtubule at that gamma ring. So the microtubules become anchored on either end, which keeps them from breaking down. They're anchored on their negative end. They're at the centrosome at the gamma ring, and they're anchored on their positive end by touching the positive end of a microtubule from the other side of the cell. Microtubule motor proteins also play a role here. They work to promote interactions between the microtubules and the formation of the spindles. By walking along these microtubules, they get the microtubules oriented properly. Now, with the mitotic spindles in place and the chromosomes replicated and condensed, the only thing that remains is attaching the sister chromatids to the spindle microtubules so that the chromatids can be pulled apart as we move from metaphase into anaphase, be pulled apart to opposite poles of the cell. So, everything we're talking about here begins in prometaphase. We're now in prometaphase, the I proudly prefer part of mitosis. Again, the hallmark feature of prometaphase is the breakdown of the nuclear envelope due to the phosphorylation of the nuclear lamins. And the pieces of the nuclear membrane become scattered as these vesicles of nuclear phospholipids. This was triggered by, again, the phosphorylation of the nuclear lamins. But what phosphorylated the nuclear lamins? The appropriate CDK, the MCDK did. Once the nuclear lamins are phosphorylated and the nuclear envelope breaks down, now the chromosomes are no longer in a nucleus. The chromosomes are liberated, and they're essentially floating in the cytoplasm. And this is critical. This has to happen before metaphase because metaphase is the spindle microtubules attached to the chromosomes and center them in the middle of the cell. Well, the spindle microtubules are being made from centrosomes that are out there in the cytoplasm. The spindle microtubules were always cytoplasmic because that's where the centrosomes were. So in order for the spindle microtubules to attach to chromosomes, we have to have no nucleus. If the nucleus was left intact, then we would have this barrier between the chromosomes and the spindles. With the nucleus gone and everything's together floating in the cytoplasm, now the microtubules of the spindle can be attached to or capture, like that fish in the pond, can be captured. The chromosomes can be captured by these microtubules. So how is this happening biochemically? Well, the way that the mitotic spindle fibers, that these microtubules attach to the chromosomes, is through a specialized protein complex called the kinetochore. And each sister chromatid has a kinetochore at its centromere. I know a lot of terms, so here's the picture. In brown, we see each of the sister chromatids. They're held together by cohesin. At the centromere, where these chromosomes contract, where they get a little bit tighter, a little bit narrower, we have the kinetochore. Kinetochore is a quaternary protein structure of many, many subunits. The kinetochore is on one end attached to the centromere, which is part of the chromosome, and on the other end attaches to mitotic spindles. So it's at the kinetochore where mitotic spindles are bridged to the sister chromatids. Because the kinetochores face in opposite directions, each sister chromatid can become linked up with a microtubule from opposite sides of the cell, from opposite centrosomes. And we now have our attachment. Now, to be fair, this picture right here is kind of late prometaphase, early metaphase, because the chromosomes there in blue are starting to line up at the center of the cell. But that lining up can only occur when we have proper attachment of the chromosomes to the mitotic spindles. This attachment of the still cohesin-connected sister chromatids to the microtubules through the kinetochore, and the microtubules from opposite centrosomes from opposite sides of the cell, creates a kind of tug of war on the sister chromatids, as each microtubule from opposing centrosomes pulls on the sister chromatids, and the sister chromatids can't give way because cohesin is still present. We get equal and opposite tension on the chromosome, and this is what allows the microtubules and the centrosome to physically line up the chromosomes at the center of the cell. In other words, if we think of the microtubules as fishing lines, those fishing lines are taut, they're tight. Initially, microtubules from opposing centrosomes are lengthened and contracted, and this pulls the chromosome back and forth as each centrosome fights for greater tension. And so the chromosome, still with two sister chromatids attached, is jerked roughly towards each spindle pole. But eventually, an equilibrium is met, almost like an evenly matched game of tug of war. The tension coming from either centrosome becomes equal. And that allows the chromosomes to end up being perfectly aligned in the center of the cell. And we call that imaginary plane through the center of the cell the metaphasic plate. Once the chromosomes are lined up single file on the metaphasic plate, we are in metaphase. That's the hallmark feature of metaphase, is the alignment of these chromosomes in the center of the cell. And this is, I don't even want to count them, many, many cells all synchronized in metaphase. The green fluorescents are the chromosomes, and the orange fluorescents are the microtubules. We can experimentally demonstrate that there is actual tension on these microtubule lines by severing the connection between one microtubule and the sister chromatid, the kinetochore that it's attached to. And when we do that experimentally, you can probably come up with a hypothesis. And I hope this was your hypothesis. Because of that tension now only coming from one centrosome, the entire sister chromatid pair is both pulled to one side of the cell. And that's exactly what we see. The entire chromosome, both chromatids, get pulled towards the microtubule that was left intact, the microtubule that was still attached to the kinetochore. So the tension of the microtubules from these opposing spindle assemblies, again through attachments at the kinetochore on different sister chromatids, is the signal to the cell that everything is proper in mitosis. This is what needs to happen in order for the mitotic checkpoint to be passed. This is the M phase checkpoint. Equal tension on both sides of a chromosome, equal tension being applied to each sister chromatid, still connected by cohesin. And that tension is due to microtubules on opposing centrosomes attached equally at the kinetochores of the two sister chromatids of a chromosome. The cell cycle control system is monitoring this tension. If that tension is not equal and present on both sister chromatids, then M phase arrests, and the cell is given more time for microtubules to attach to those sister chromatids. If we don't pause the cell cycle right here, then this can happen. Here we see the blue chromosome has equal tension on both sides. Microtubules have attached to each kinetochore of each sister chromatid of the blue chromosome. And as we progress onto anaphase, the blue chromosome will be separated properly. But the red chromosome had the microtubule on the right miss. The centrosome projecting the microtubule on the right didn't connect to the kinetochore, while the centrosome projecting the microtubule on the left did connect. And so if we were to pull, we're going to pull both sister chromatids in the left direction. And the end result of that will be a cell with one chromosome too few on the right and a cell with one chromosome too many on the left, and that is aneuploidy. That state of the wrong number of chromosomes is referred to as aneuploidy. Once all chromosomes are correctly oriented at the metaphasic plate, and there's equal tension across all chromosomes and all sister chromatids are attached to microtubules, the cell has successfully completed metaphase and can now move on to anaphase. Now the sister chromatids must be separated and pulled to opposite sides of the cell, and that's exactly what anaphase is for. So what allows that to occur from a cell biology perspective? Well, the beginning of anaphase is marked by the release of cohesin linkages. Cohesin needs to be destroyed so that sister chromatids can be physically pulled apart. Remember, cohesin is the glue or the rings that were keeping the sister chromatids bundled. So step one of anaphase is to get rid of cohesin. And we can go from metaphase on the left to anaphase on the right. And sister chromatids are physically separated from each other, becoming independent chromosomes. That'll happen when the spindle microtubules keep pulling with their tension. They're still playing tug of war equally there, but as cohesin is destroyed, now those sister chromatids give way and they can be pulled apart. The process of these sister chromatids being pulled apart is the law of segregation, Mendel's law of segregation. The protein that's responsible for destroying cohesin so that all of this can occur is called separase, which is a pretty crazy name for an enzyme. It's like flipase and scramblase. Separase allows sister chromatids to be separated. Separase is activated by phosphorylation. Actually, it's activated by a complex of proteins, which also targets mCDK for destruction. Because right now we're about to enter anaphase. We're getting close to the end of mitosis, so it's time to start wrapping mitosis down, which means it's time to start getting rid of the mCDK because we're about to finish mitosis. This protein complex that turns separase on and simultaneously destroys MCDK is called the anaphase promoting complex, the APC. Good name for it, because this promotes anaphase. Thus, the beginning of the end of mitosis has just been initiated, and that's what the anaphase promoting complex does. It begins the end of mitosis. We've made it through metaphase. We're deep into anaphase now. We've got telophase, and we're done. And so the APC is activated. The APC activates separase so that cohesin can be destroyed. The cytochromatids just come apart now because they already had tension upon them. So we immediately go into anaphase as soon as cohesin is destroyed. And that means we're successfully in anaphase. It's time to start wrapping up mitosis. It's time to destroy the MCDK. So the APC does two things simultaneously. Once we've made it to anaphase, once we have passed the M phase checkpoint, and that is one, destroying cohesin through separase, and two, destroying the MCDK, so that we begin to transition to the end of mitosis. Chromosomes are separated, they're pulled apart, and the MCDK is destroyed, both being triggered by this APC anaphase promoting complex. Okay, so chromosomes are pulled apart during anaphase. Cystochromatids separating and being pulled to opposite poles of the cell results in two independent processes occurring. It's anaphase A and anaphase B. Anaphase A involves the microtubules being attached to the kinetochore and being actively shortened by depolymerization of the microtubule. And this drags the chromosome to one pole. And that's anaphase A. Again, the shortening of microtubules. And as microtubules shorten, we're reeling in the cystochromatid. In anaphase B, motor proteins walk the spindle poles themselves further and further towards the pole of the cell. So anaphase B is the physical movement of essentially the centrosomes further and further apart to more and more extreme ends of the cell. And this also pulls the chromosomes further apart. So anaphase A and anaphase B happen simultaneously, and together they work to rapidly get cystochromatids from the center of the cell to the poles. The driving force for anaphase A appears to be also motor proteins that are attached to the microtubules right adjacent to the kinetochore. And these motor proteins use energy of ATP hydrolysis to actively kick alpha and beta tubulin dimers away from the plus end of the microtubule. So we're shortening the microtubule at the plus end. This is an active disassembly mechanism. This isn't dynamic instability, where the microtubules, the tubulins dimers didn't really want to be there and they just fell apart. This is energy being expended to rapidly dissociate the microtubule. Dynein and kinesin, our old friends, our old motor proteins, play a role at the spindle poles, pulling those centrosomes further and further apart for anaphase B. It's believed that these motor proteins are associated with different microtubules that are part of like an ad hoc cell cortex, but we're really not sure exactly how that works still, or at least not to my knowledge. Perhaps in the last couple of years that's been determined. By walking on these microtubules, the motor proteins are essentially dragging the centrosomes closer and closer to the cell membrane, or in other words, further and further apart from each other, and this is pulling the spindle poles and the cystochromatids those microtubules are attached to further apart. And as you can probably tell from the language I'm using here, I keep saying it appears, we believe, we really don't understand this process in any depth. And this is where new PhD projects come from. So for those of you that are interested in graduate school, this is the kind of loose end that becomes a PhD. What's going on here? Let's figure it out. Okay, so before we move on from this, I just want to use an analogy here. If we think of anaphase A and anaphase B occurring simultaneously, for me, the simplest analogy to think about this is indeed someone fishing. So imagine a person is standing on the shoreline of a pond or a lake and they're fishing. So they are the centrosome. And the fishing line that they've cast into the pond is the microtubule. And they caught a fish. And that fish is a cystochromatid. Now, that fisher person, fisherman, fisherwoman, can get that fish to shore in two different ways. They can start reeling in the fishing line to make the fishing line shorter. That's going to get the fish towards the shore. Or they can start walking backwards, and that will also drag the fish out of the lake. So they can reel in the line, make the fishing line shorter, or walk backwards. And if they want to be extremely expeditious, they can do both. They can reel that line in and walk backwards, and that fish will come out of the lake as fast as it can. That's exactly what's going on in the cell here. The reeling in of the fishing line is anaphase A, and the walking backwards of the fisher person is anaphase B. And the cell does both simultaneously to get that fish of a cystochromatid to the cell pole as quickly as possible. So we already saw the consequences of anaphase occurring before all cystochromatids were connected. There was that aneuploidy example. One pole would receive both cystochromatids and the other would receive neither. Again, this is called aneuploidy, an inappropriate number of chromosomes, one too few or one too many, in the daughter cells. Usually, aneuploidy is lethal to the cell. In fact, the only aneuploidy that cells can tolerate is aneuploidy 18, 21, and of the X chromosome. The reason for this is that microtubules attach to all kinetochores in the cell cycle, and that's the checkpoint being checked for. That's the M phase checkpoint, making sure that these microtubules are attached so that aneuploidy will not occur. We want the scenario on the right, equal tension across all chromosomes, not the situation on the left where one microtubule has missed. There's no tension on that topmost chromosome, and now the cell on the right is going to get one chromosome too many, and the cell on the left is going to get one chromosome too few. Even a single case of a missed attachment should result in the arrest of mitosis to give the cell time to attach all kinetochores, and if it can't, then to go into apoptosis. So what's actually manifesting this checkpoint? The APC. The APC is what is involved in this checkpoint. If there is not equal tension across these chromosomes, the APC will be blocked, will be kept inactive. And without an active APC, cohesin will not be degraded, cystochromatids will remain separated, and anaphase cannot be progressed into. So again, I know I made the point on the previous slide, but the determining fact, I should say the determining action that occurs when the M phase checkpoint is passed is the activation of the APC. And that gets you moving from metaphase into anaphase. Likewise, as we don't have equal tension on that chromosome and kinetochores have not been successfully attached to by microtubules, the APC is kept inactive, it's kept dormant. And then we remain stuck in metaphase and we don't progress into anaphase. So all of this kind of comes together biochemically or from a cell biology perspective. So in prometaphase, we're just starting to attach to chromosomes. We don't expect all chromosomes to be attached. That's not the goal of prometaphase. We're just getting everybody situated. Then in metaphase, remember, we shorten and lengthen, shorten and lengthen. We tug on either side with these centrosomes, looking for equal tension. And then when equal tension is sensed across all chromosomes, then the APC can be activated. It's allowed to activate. The APC activates. Two things happen. Cohesin is destroyed, and now that tension allows the cystochromatids to be pulled apart. And simultaneously, we destroy the MCDK, allowing us to begin our exit from mitosis. So by keeping the APC inactive when we fail the metaphase checkpoint, the cell is given time to try to reattach their microtubules to all kinetochores, making sure that we achieve tension on all of these microtubule lines. This is called the M phase checkpoint informally. This can be called the metaphase checkpoint, but formally this is called the spindle assembly checkpoint because that's exactly what it's a checkpoint for the full assembly of the spindle. Okay, so everything happened properly. Cohesin was destroyed. Cystochromatids were separated. They're now referred to as chromosomes, and they're on opposite sides of the cell. And we've broken down the MCDK. APC has done its job, and each side of the cell has a complete complement of chromosomes, has a complete genome. Now we have to enter telophase. So after all of that has occurred, the microtubules can disassemble. Their job is done. The chromosomes have been pulled to opposite sides of the cell. It's actually time to start building the microtubule tracks that we see in interphase. We have to enter telophase now, which is the final stage of mitosis. And telophase is marked by the formation of new nuclei around those genomes on opposite sides of the cell. This is going to occur through the dephosphorylation of the nuclear lamins. Those little vesicles of scattered nuclear phospholipids of nuclear membrane that we left there in prophase when the original nucleus was disintegrated, they're all going to cluster around the segregated chromosomes, and those vesicles will start to fuse to form more nuclei membrane, and then that nuclear membrane on either pole of the cell needs some structure, needs some cortex under it to give it its shape and its integrity, and that's going to require the nuclear lamins. The nuclear lamins are going to be dephosphorylated so they reassemble, and nuclear pore complexes will be embedded in the gaps of that double membrane. So it's this beautiful cycle of phosphorylation and dephosphorylation. Phosphorylation in prometaphase to destroy that nuclear envelope, and then we keep those lipids around, we keep those membrane components around, they're just unable to form a structure, and then we dephosphorylate the nuclear lamins, allowing that membrane to reform at the middle of telophase. So the nuclear envelope reforms, the nuclear cortex reforms with those intermediate filaments, the pores become active, and so they start allowing in the nuclear proteins that need to be in the nucleus. All those nuclear transport receptors are carrying in the nuclear proteins. There's also ways to get material that wound up in the nucleus that shouldn't be there out. The chromosomes decondense now. They go back to their more linear form, and gene expression can resume. The cell has left its mitotic behavior because the MCDK was destroyed, and the cell is now resuming its normal behavior. In fact, the only thing that we haven't done is split that one cell into two. So mitosis is over, and it is now time for cytokinesis. We now have two complete nuclei, but they exist in one cell. We have to split this one cell into two to fully complete M phase and to complete the cell cycle. Once cytokinesis occurs, M phase is over and the cell cycle is over as well. And the groundwork for cytokinesis was actually laid in Anaphase. The cell splitting in half is fairly complicated, and all of the proteins and processes that are responsible for cytokinesis had to be prepared. And so that preparation began in Anaphase. The most critical component for cytokinesis is the actin contractile ring that we've been talking about for multiple lectures now. The ring itself is made up of actin, and then myosin is going to be involved to get that ring to contract. Where this ring assembles and where the cell actually splits is determined by the mitotic spindle. We call this the plane of cleavage. So what? The mitotic spindle, the microtubules, dictate where the actin forms and where the cell is going to split? Yes, that's what we're saying. During anaphase, the cell membrane begins to take on a kind of a pinched in or furrowed appearance, a puckered appearance, in a line that always runs perpendicular to the direction of the mitotic spindle. And this is what we're talking about. This is that furrowed or ruffled appearance. And so if this here is the plane or direction of the spindle microtubules from left to right, this will be the plane or direction of the cleavage furrow, up to down. The cleavage furrow always forms perpendicular to the plane of the microtubules, always in this way. And that makes so much sense, right? Because if this centrosome is here on the left and this centrosome is here on the right, that means when anaphase goes to completion, one set of chromosomes is going to end up on the left and one set of chromosomes is going to end up in the right. So you damn well better split that cell down the middle so that the cell has, each new daughter cell has its own genome. This positioning ensures that when the cell splits along this furrow, we're going to have a set of chromosomes in each new cell. If the spindles are if the spindles are experimentally and intentionally displaced, so we do something like this, then we see that the furrow appears in a new position as well. still perpendicular to that new plane of microtubules. So it is definitely causal that the plane of the microtubules is directly informing the cleavage furrow. How the spindles orient places the cleavage furrow where it goes. Why and how that happens? There's another PhD project. Don't know. Don't know. Usually, the mitotic spindle is centered in the cell. And so this usually results in a cleavage furrow that is also centered in the cell and just perpendicular to the mitotic spindles. And the cell will usually be evenly divided into two roughly equal sized cells. Sometimes, though, especially during development, it's purposeful for a cell to have an asymmetric division. This is especially true when females are making egg cells, oocytes. We want the actual egg to be as big as possible and contain cytoplasm and organelles and everything else the cell needs to be sufficient. And we want the other daughter cell to be as small as possible so it doesn't lose that stuff. So there are times where a cell actively seeks an asymmetrical division. Interestingly, beautifully, this asymmetrical division is accomplished by moving the spindle assembly to a non-centered position. When the spindle assembly is non-centered, the cleavage furrow is non-centered. The cleavage furrow always follows the plane of the spindle. And so the cell on the left with its unequally shared spindle apparatus is going to become the cell on the right with its asymmetrical cleavage furrow. Cool, right? Amazing. And so, of course, we'll get two daughter cells that are different sizes. Many times, the two different daughter cells also contain different molecules. They differentiate into different cell types. For the oogenesis story, when we're making eggs, the other cell is actually called a polar body and it rapidly dies. So this is all intentional when it's done this is all intentional when it's done properly for specific cells. This is one way by which cellular differentiation during development can be achieved. Okay, so let's talk about that cleavage furrow and the contractile ring itself. Very much like muscles, like the sarcomeres that we talked about in the cytoskeleton lecture, the contractile ring is made up of interlocking actin filaments and myosin holding onto it. Except unlike a muscle where it's all linear, here it's arranged in a ring. The contractile ring is assembled in anaphase, as I mentioned before, and it's attached to the cell membrane. Well, that's easy to imagine because it's actin. And actin always attaches to the cell membrane when actin is part of the cell cortex. So actin is able to attach to the cell membrane. The actin binding proteins allow that. Unbelievably, the contractile ring can generate enough force to bend a glass needle. This was shown experimentally. So what's actually happening here is if the blue is the ring of actin and there's myosin embedded in those actin fibers, as the myosin goes through the power stroke, and remember what the power stroke is, ATP, cleave to ADP and phosphate, and then release the phosphate. ATP, cleave to ADP and phosphate, and then release the phosphate. That is release, bind, pull. Release, bind, pull. Release, bind, pull, release, bind, pull. And as the myosin release binds and pulls the ring, the diameter of the ring is going to get less and less. And since the ring was attached to the cell membrane is going to be pinched inward. And it's going to be pinched inward with so much force that that one cell will be physically pinched into two separate cells by the power of this contractile ring. The contractile ring is almost like a belt being cinched to its utmost, forcing that cell to split into two. Once the cell is completely pinched, the two cells kind of bud away from each other, very much like a vesicle being pinched off of the bud neck there by dynamite. We talked about that before. And that one mother cell becomes two daughter cells. At that point, the contractile ring is no longer needed. It disassembles. It was a transient structure to begin with, and the actin can be remade into the cell cortex and all the other actin-containing structures that the cell needs. All right, so that's pretty much the story. I just want to wrap up here with a few side stories to give you a sense of some particulars of how this works. As I mentioned in the intro for this video, we're going to talk a little bit about plants here, and then we'll talk about how cells control their cell number. So plants are unique from animal cells in that plants have a cell wall. And so it's certainly not enough to just pinch the membrane apart. We need to build walls to create two cells from one. So throw all of that cytokinesis stuff away when we're dealing with plant cells, because what happens in plant cells is completely different. Now, to be fair, plant cells perform mitosis almost identical to any animal cell. It's just that they perform cytokinesis very, very differently. So from prophase to telophase, all of that is basically the same. Plant cells are not flexible in the way animal cells are because plant cells are characterized by having a cellulose cell wall. And so plant cells can't be pinched apart. They need to be bricked apart. A new cell wall has to be built in the one mother cell to split that one mother cell into two dividing cells. And that new cell wall begins being made in telophase. Small vesicles filled with cellulose and other complex sugars and glycoproteins, the main constituents of the cell wall, are transported to the center of the plant cell. Again, they're vesicles, so they're being transported by motor proteins, walked along microtubules. We know how that works. Then, in that metaphasic plate of the plant cell, those vesicles are going to fuse to form a membrane, and the contents of all of those vesicles will also integrate and become an enclosed disc. This is an enclosed disc of complex polysaccharides. Once this disc, this membrane-bound disc of polysaccharides, gets so big from all of this fusion that it extends out to the wall of that mother cell, the membranes are going to fuse with the cell membrane. That disc is going to be liberated between those cell membranes, and you've got yourself a cell wall. Here it is. Here it is. So here are the vesicles being shipped to the metaphasic plate, to the center of the cell. Now, the chromosomes aren't there anymore because they've already been pulled apart to opposite poles. And these membranes contain cellulose and the other building blocks of the cell wall. And these membranes start to fuse. And as the membranes fuse, the membrane is in red. As the membranes fuse, the vesicles essentially get bigger and bigger, and the interior components of the vesicles also integrate. So now here we see the vesicles are getting even bigger still, and so too is the complex carbohydrate in them, until ultimately, again, membrane is in red. Well, this is membrane in red, and cell wall stuff is in yellow. Now the membrane of the vesicle becomes the membrane of the cell. The contents of the vesicle become this middle cell wall between the cells, and one plant cell has been turned into two. Later on, this kind of holder wall will become, with higher integrity, it'll become more durable as cellulose fibers are added to it, completing it and making it whole. The position of the new cell wall affects the overall structure of the plant, and so this positioning is very precisely controlled. What about the organelles? How do we get the right organelles to daughter cells as those daughter cells are dividing? So at this point, we've covered mitosis, getting chromosomes to new nuclei. We've talked about cytokinesis, turning one cell into two. We've talked about cytokinesis in plants, turning one plant cell into two. But what about the mitochondria? What about the Golgi? What about the ER? No organelle in a cell can be made from scratch, so how does each daughter cell get its set of organelles from the mother cell? Well, actually, for mitochondria and chloroplasts, it's easy, since there's thousands and thousands of them in every single cell, for one. And since they're essentially prokaryotes and they can replicate themselves, for two, there's always going to be at least some mitochondria and chloroplasts in the daughter cells. And so the cell doesn't really go to too much trouble to make sure that it's an equal amount. It's just that each daughter cell has to get some of these organelles, and then they can repropagate and reconstitute themselves through the prokaryotic cell division that they're capable of. Not so for the ER and the Golgi. The way that we make sure that the daughter cells get ER and Golgi is that the ER is kept near the center of the cell. The ER is centered along the metaphasic plate where the nucleus used to be. And remember, the ER was always contiguous with the nucleus. So when cytokinesis occurs, cytokinesis is occurring right through the ER. And cytokinesis quite literally cuts the ER in half. So each daughter cell gets a half ER. And then with phospholipid regeneration, that ER can grow in size. Also, what happened during M phase, earlier on in M phase, is that the Golgi layers intentionally fragmented into even smaller vesicles. So the Golgi apparatus becomes just many, many, many small vesicles. And these vesicles become attached to motor proteins, motor proteins that are able to walk along the spindles of the microtubules. And so as the sister chromatids are being pulled to opposite poles of the cell, these Golgi vesicles hitch a ride along with them and also get pulled to opposite sides of the cell. You can think of this as seaweed getting caught in the fisherman's line. So as the fisherman is reeling in the line and walking backwards to bring that fish to shore, the fisherman is also bringing seaweed to shore unintentionally. And in that analogy, the Golgi vesicles are the seaweed. Pretty cool there too, how everything finds its way to end up in the daughter cells. Finally now, what about the controlling of cell number? And we'll be very brief about this. This is essentially a lead up to cancer, and cancer is our last lecture. And so we're just laying some groundwork here to think about cells in communities, which is what our last week is about. Cell division is critically important, no doubt about it. Multicellular life in fact itself, is not possible without the cell cycle and cell division. So no argument there. But for a multicellular organism, not dividing can be just as important as the ability to divide. Cancer is a disease of uncontrolled cell growth that's caused by accumulated mutation, and cancer can be fatal. A yeast cell dividing uncontrollably when nutrients are not plentiful in its environment just created a whole lot of competitors for the resources that were already in limited supply. And so it is important for cells to know when they should be dividing. It is equally important for cells to know when they should not be dividing. And that decision point is one of the G1 checkpoints. When we talk about environmental cues, this is what we mean. We need a mechanism that tells cells when it's appropriate to divide. This is beyond checkpoints. This is controlling entry into the cell cycle itself. We also ideally would need a go-back mechanism where extra cells can be gotten rid of just in case we've made too many. And this one is easy to address. The go-back mechanism for a multicellular organism is apoptosis, pre-programmed cell death, or sometimes referred to as cell suicide. All cells are born with a genetic program by which they can use to end their own existence. This is a type of cellular autodestruct, and again, it's referred to as apoptosis. During development, it's very common for the body to make too many cells. In fact, it's often done on purpose. This is especially true in the brain and the central nervous system. Many more neurons are made early on than we could ever use, than we ever need, than can ever connect to each other. The same is true for skin. And then with these initial cells in place, essentially the developmental programming and genetics of the organism sculpts the organism into the appropriate structure by trimming away the excess of these cells, just like you would trim away the excess clay of a sculpture you were trying to make. The way this sculpting is accomplished is by apoptosis. Cells that are deemed unneeded are launched into apoptosis and die, and the cells that would be the clay we're cutting away, and the cells that we want to keep are not allowed to go into apoptosis. So apoptosis is responsible for the separation of fingers in all mammals. All of our hands began like this, more of a paddle. To be clear, the bones and the musculature under that paddle of skin was articulated, but the skin itself was one solid mitten over those bones. That's too much skin cells. And then over time, with apoptosis, the skin cells between the fingers were killed off, were allowed to die, separating the fingers from each other. Sometimes this apoptotic program doesn't work quite well, and an individual can be born with their fingers fused together. This is just the ineffective apoptosis programming of the skin cells between these two middle fingers. Very easily fixed with surgery, not a long-term disability, but an example of how this process can go wrongly. Neuron number is reduced in very much the same way. Apoptosis reduces neuronal number in the brain tremendously, and this allows the proper and correct connections in the brain to be maintained and the improper, incorrect connections of the brain to be lost. This fine-tunes the functioning of the brain. And this is all happening through chemical cues. This is happening because when a neuron successfully connects to a postsynaptic neuron, the postsynaptic neuron releases chemicals, paracrine chemicals, that tell the presynaptic neuron, you have connected successfully, stick around, we've got a good connection. Other neurons that connect to neurons inappropriately don't get those signals and wither away and die. They undergo apoptosis. In fact, even for a frog's morphogenesis, this is what's happening. What causes a tadpole's tail to disappear as it metamorphs into a frog is apoptosis. All good things. Separating the webbing between our fingers, good thing. Fine-tuning the brain, good thing. A frog not having a tail, good thing. Apoptosis is a good thing when it occurs appropriately. And so it's extracellular signals that allow appropriate cells to continue surviving, continue growing, and continue dividing. But there are additional levels of regulation for cell division, and most of them rely on cell signaling mechanisms. Nice tie-in to a couple weeks ago. Multicellular organisms are communities. They are societies of cells. And just like functioning societies and functioning communities, these cells have to communicate with each other, they have to coordinate their activities, and they have to follow the rules. And so cellular signals called survival factors reward the cells that are following the rules by blocking the apoptosis in those cells. Apoptosis is actually often the default pathway. Cells will automatically self-destruct if they're not told not to by survival factors. So what happens is a survival factor binds to a receptor. The survival factor is the primary messenger. The receptor transduces that signal into the cell. The secondary messenger is a transcription factor that enters the nucleus and binds to a gene. And then a protein is made that blocks apoptosis. This cell was told to not self-destruct. It's doing a good job. It's needed in the community, and it should stick around. For cells that are inappropriately behaving, neurons that misconnect to other neurons, skin cells that are making up the webbing between fingers, the survival factor will not be given to that cell. This apoptosis blocking protein will never be made, and the cell defaults into apoptosis and destroys itself. Cellular signals called mitogens promote mitosis by helping cells to relieve the G1S checkpoint block. Again, not progressing through the cell cycle is the default state. So you see this. Default state is to self-destruct. Default state is to arrest in the cell cycle. Growing and dividing is the activated process. Same similar thing. Mitogen is the primary messenger. The mitogen binds to a receptor at the cell cycle, at the cell surface, and we get signal transduction into the cell. Here, the signal transduction allows the G1S boundary CDK to become active. It relieves the inhibition of the G1S CDK. And when we relieve that inhibition and the G1S CDK can become active, we progress from G1 to S phase and we enter the cell cycle proper. Other cellular signals called growth factors promote cell growth. They actually cause the cell to increase in size by increasing the production of cellular membrane and other cellular components. Getting sick of it yet? The growth factor is the primary messenger. It binds to a growth factor receptor at the cell membrane. Signal transduction allows that information to enter the cell through secondary messengers, and the end result of that is 1, protein and lipid synthesis is increased, and 2, other proteins that would cause apoptosis are degraded, and the cell survives to grow. See what these all have in common? It's all signal transduction. It's all signal transduction. It's all signaling cascades. Cell biology is truly one giant interconnected story. What happens when these receptors misbehave? What happens when these receptors transduce a signal even in the absence of a primary messenger? What happens when the proteins responsible for apoptosis are mutated and apoptosis cannot occur? What happens when everything we talked about in these last 2 lectures goes wrong? Cancer happens. And to understand cancer, which is our last lecture, we have to understand how cells behave in communities. And that's our next lecture. So 2 more lectures to go, folks. Lectures on tissue formation and cellular communities, and then the lecture on cancer, and we're done. What did we talk about here? Well, we talked about mitosis. For chromosome segregation in anaphase, first we have to have a duplicated centrosome, and that's going to happen all the way in prophase. Then microtubules can grow out of each of those centrosomes on opposite poles of the cell, and they touch their positive ends in the middle, and this forms the spindle apparatus. The centrosomes, the microtubules of the coming out of the centrosomes are stabilized through this touching. Along the way, the nuclear envelope is breaking down due to the phosphorylation of the nuclear lamins, and the nuclear membrane forms these little vesicles. That's happening in prometaphase. The spindle microtubules can now capture or attach to sister chromatids through these kinetochore protein complexes. And then by shortening and lengthening, by tugging and pulling, we can get those chromosomes all lined up at the metaphasic plate. And once we do, we are in metaphase. The tension of these microtubules by attaching to the kinetochores on opposite sister chromatids is the M phase checkpoint. It's the spindle apparatus checkpoint, and we need equal tension on both kinetochores in order for the cell to progress from metaphase into anaphase. What allows that progression? The activation of the anaphase promoting complex. Once the APC is activated because we've passed the M phase checkpoint, separase is going to be turned on, and separase is going to chew up cohesin and allow those sister chromatids to be physically pulled to opposite sides of the cell. What mechanisms are allowing those sister chromatids to be pulled to opposite sides of the cell? Well, the fisherman reeling in the line, active motor proteins breaking down the positive end of a microtubule, that's anaphase A, causing that microtubule to shorten, and the fisherman walking backwards from the shoreline, pulling that fish to shore, that's anaphase B. Motor proteins walking centrosomes closer and closer to the cell membrane, getting them further and further apart. All of that being triggered by the APC. After sister chromatid segregation has completed, the spindle microtubules are going to disassemble due to dynamic instability, and we're going to dephosphorylate the nuclear lamins, allowing those nuclear membrane components to come back together, forming two new nuclei around each of those genomes. And we are now in telophase, the final stage of mitosis. With mitosis over, we can enter cytokinesis. Cytokinesis was actually started in anaphase as the cell cortex was broken down and those actin filaments were reassembled into a contractile ring. And so cytokinesis overlaps mitosis. But once we're done with telophase, all that's happening is cytokinesis, and that's the contractile ring being tightened. The contractile ring is being contracted through myosin pulling on actin filaments via the power stroke. Release, bind, pull, release, bind, pull. Myosin pulls this actin filament in until the diameter of the contractile ring gets smaller and smaller and smaller, and eventually the contractile ring will pinch that one cell into two, and cytokinesis is done. We talked briefly about the importance of apoptosis and cell signaling through mitogens and growth factors to make sure that cell numbers are kept correct. Apoptosis kills off extra cells. It's the default state. And things like mitogens and growth factors tell cells to survive and grow. And it's these dueling things, killing the cells we don't need and rewarding the cells that we need by allowing them to live and grow, that keeps a multicellular organism healthy. And if these processes are disrupted, we will have too many cells or too few cells, and disease states will occur. And that's exactly where we're going to go next week. So please, as always, let me know what from this video you would like to review in the classroom. And I can't say it enough. Thank you so much for your attention and for watching this video.